Good day everyone, today we'll be discussing gene mutations. So a mutation is a change in gene's nucleotide base sequence. So it can be substituting one DNA base for another or deleting a few bases at the chromosome level. So chromosomes can exchange parts, genetic materials can even jump from one chromosome to another. Effects of mutation. So mutations can happen regularly. It can be caused by chemicals and UV radiations. Many mutations are repaired by enzymes. Some type of skin cancers and leukemia result from somatic mutations. So mutations may be present in all cells of an individual or in just some cells. In a germline mutation, the change occurs during the DNA replication that precedes meiosis. While in somatic mutation naman, the change happens during DNA replication before the mitotic cell division. So all cells that descend from original changed cell are altered, but they might only comprise a small part of the body. Somatic mutations are responsible for certain cancers. So beta globin gene. So the first genetic illness to be understood at the molecular level was sickle cell. So Ernest Irons noted many pear-shaped, elongated forms in a blood from a dental student in Chicago who had an anemia. So Irons sketched this first view of sickle cell disease at cellular level and reported his findings to his supervisors, James Herrick. So Herrick published the work but did not mention Irons. So Herrick has been credited with the discovery ever since. So this is the normal cell. Discoid shapes, unlike sickle cell, they are like pear-shaped blood cells. And then Linus Pauling discovered that hemoglobin from healthy people and from people with anemia, when placed in a solution in an electrically charged field, move to different positions. So the difference between the types of hemoglobin lay in the beta globin. VM in gram identified the tiny mutation responsible for the sickle cell disease. It is the substitution of amino acid valine for glutamic acid that is normally in the sixth amino acid in the beta globin polypeptide chain. So the aggregated hemoglobin molecules form rope-like cables that bend red blood cells into rigid, fragile, sickle-shaped structures. The mishapen cells lodge in the narrow blood vessels, cutting off local blood supplies. So once blockage occurs, sickling speeds up and spreads as the oxygen falls. So it causes great fatigue of anemia. So sickle cell disease was the first inherited illness linked to a molecular abnormality, but it was not first known condition that results from mutation in the beta globin. Thomas Cooley and Perley describe severe anemia in Italian children. So, ang tawag dun is Cooley's anemia. The disease was named as thalassemia from the Greek word "sea" because it is highly prevalent in the Mediterranean area. The severe form, sometimes called thalassemia major, results from homozygous mutation in the beta globin gene. The milder form has the thalassemia minor, affects some individuals who are heterozygous for the mutation. The disorder that is more common in the Mediterranean is more accurately called beta thalassemia. Without them, not enough hemoglobin molecules are assembled to efficiently deliver oxygen to the tissues, so fatigue and body pain may arise. So, as this progresses, the RBC dies because the excess alpha globin chains prevents formation of the hemoglobin molecules. So, liberated iron slowly destroys the heart, the liver, and the endocrine glands. Drugs called chelators that entrap the iron can extend life past early adulthood, but they are very costly and not available in developing nations. Disorders of orderly collagen. So collagen is a major component of connective tissue. So it accounts for more than 60% of protein in the bone and cartilage and 50 to 90% in dry weight skin. So it can be also found in eyes and blood vessel linings. So what are the different collagen disorders? First, we have the Alport syndrome, mutation in the type 4 collagen, and it can cause deafness and inflamed kidneys. Aortic aneurysm, a missense mutation, substitute arginine for glycine in the alpha-1 gene. It can cause aorta burst. Chondrodysplasia, deletion, insertion, and missense mutation replaces glycine with bulk amino acid. It can cause standard growth and deformed joints. Dystrophic epidermolysis bullosa, collagen fibers that attach epidermis to dermis break down. It can cause skin blisters on any type. We also have Ehlers-Danlos syndrome. Missense mutation replaces glycine with bulk amino acids. Can cause stretchy, easily scarred skin, lax joints. We have osteoarthritis, substitute cysteine for arginine in the alpha-1 gene and can cause painful joints. We also have osteogenesis imperfecta type 1, inactivation of alpha allele reduces collagen triple helices by 50%. It can cause easily broken bones, blue eye, whites, and deafness. We also have Stickler syndrome, nonsense mutation in procollagen. It can cause a joint pain, degeneration of vitreous gel, and retina. So that is the picture of those individuals with dystrophic epidermolysis bullosa. So early onset of Alzheimer's disease. The discovery of a mutation that causes an early onset autosomal dominant form of Alzheimer's disease began when a woman named Hannah developed progressive dementia in her 40s. Then many of her descendants also experienced dementias, some as early as their 30s. Then Hannah's grandson, they're both physicians, conducted an extensive pedigree tracing Alzheimer's disease in their family to identify their family's. Then they narrowed the investigation to a portion of chromosome 14 and pinpointed the gene 3 years later. It encodes a protein called presenilin 1 that acts as receptor anchored in the membrane of Golgi apparatus or a vesicle. So the protein monitors the cell storage or use a beta amyloid. It is a substance that accumulates in the brain of people with Alzheimer's. The abnormality in the presenilin disrupts amyloid production, folding, or function. So we also have multiple mutations cause confusion. So mutations in one gene may cause differing degrees of same syndrome, whereas other genes, different mutations may be associated with distinct disorder. We have cystic fibrosis associated with a gene with many mutations but one diagnosis. A man whose only symptom is infertility and occasional respiratory infection could have the same CF diagnosis as an extremely ill teenager with near constant lung infection and severe malnutrition. So mutations in the beta-globin gene cause clinically distinct illnesses such as sickle cell disease or beta thalassemia. Mutation in lamin A gene causes rapid aging disorder in Hutchinson-Gilford progeria syndrome, including muscular dystrophies and conditions. So how mutations cause disease? Example is cystic fibrosis. The protein is a cystic fibrosis transmembrane regulator, and genetic defect, missing amino acids, and the water enters the cell, drying out secretions. So frequent lung infection and pancreatic insufficiencies may be some of the signs and symptoms. We also have Duchenne muscular dystrophy. Protein involves the dystrophin, so deletion eliminates the dystrophin, and some signs and symptoms, gradual loss of muscle function. We also have familial hypercholesterolemia. The protein involves the LDL receptor, so deficient ang LDL receptor that can cause high blood cholesterol and early heart disease. We also have hemophilia A, protein involved factor 8, so absent or deficient of clotting factor, and can cause slow or absent blood clotting. Huntington disease, protein involved is the huntingtin, so extra abscess in the gene add amino acids to the protein product and can cause uncontrollable movements and personality changes. We also have Marfan syndrome. Protein involved is the fibrillin, so deficient connective tissue protein. So it can cause long limbs, second aorta, spindly fingers, sunken chest, lens dislocation. We also have neurofibromatosis type 1. Protein involved is neurofibromin, defect in a protein that normally suppresses activity of a gene that causes cell division. It can cause benign tumors of the nervous tissue beneath the skin. We also have spontaneous mutation, usually originates as an error in the DNA replication. So, characteristic of a gene is more likely to occur in repeated or symmetrical DNA sequences. Mutagens are chemicals or forms of radiation that can induce mutation by deleting, substituting, or adding bases. So mutation rates of some genes that cause inherited diseases will have Duchenne muscular dystrophy mutations per million gametes, 40 to 100, hemophilia A 30 to 60, and hemophilia B 0.5 to 10. Achondroplasia mutations per million gametes, we have 10, then very short stature, aniridia 2.6 absence of iris, Huntington disease less than 1, Marfan syndrome is 4 to 6, neurofibromatosis 40 to 100, osteogenesis imperfecta 10, it can easily broken the bones, polycystic kidney disease 60 to 120, benign growths in the kidney, and retinoblastoma 5 to 12, malignant tumor in the retina. So commonly encountered mutagens, you have aflatoxin B, fungi growing on peanuts. Then we have 2-amino 5-nitrophenol, hair dye components, 2,4-diamino-aminosol, 2,5-diamino-anisole, 2,4-diamino toluene, and phenylene diamine. All of them are for hair dye components. We also have furylfuramides, it is a food additive. Nitrosamines, pesticides, herbicides in cigarette smoke, proflavin, antiseptic in veterinary medicine, sodium nitrite, found in smoked meat. We have tris, 2,3, dibromophenol phosphate is a flame retardant in children's sleepwear. We also have point mutation, a change in a single DNA base, so it may be transition if a purine replaces a purine or a pyrimidine replaces a pyrimidine. It may also be a transversion if purine replaces pyrimidine or vice versa. So missense mutation changes a codon that normally specifies a particular amino acid into one that codes a different amino acid. So example of point mutation in sickle cell disease. A point mutation in sickle cell disease is an example of missense mutation. Nonsense mutation is a mutation that changes a codon, specifying an amino acid into a stop codon. A premature stop codon shortens the protein product. Example of that is the factor 11 deficiency. A blood clotting is a nonsense mutation, signifying. We also have a splice mutation, a mutation that can greatly affect genes product if it alters a site where introns are normally removed from mRNA called splice site mutation. It can affect the phenotype if an intron is translated into amino acids or if an exon is skipped instead of being translated, shortening the protein. So an intron is a base sequences within a gene that are transcribed but are excised from the mRNA before translation into protein. Exon, part of the gene that encodes amino acids. So these are the exons and these are the introns. So the splicing, deletions and insertions. So adding or deleting number of bases that is not multiple of 3 devastates a gene function. It disrupts the gene's reading frame. Such change that alters the reading frame is called frame shift mutation. So deletion mutation removes genetic material. Insertion mutation adds DNA and can also offset a gene's reading frame. So example, in one form of Gaucher's disease, an inserted single DNA base prevents production of enzyme that normally breaks down glycolipids in liposomes. Conditional mutations affect the phenotype only under certain conditions. Examples is common variant of the X-linked gene that encodes G6PD, an enzyme that immature RBCs use to extract energy from glucose. Unusual conditions trigger life-threatening hemolytic anemia associated with G6PD deficiency.